pleasure now to introduce Professor Waseem Qasim, pediatric immunologist and professor of cell and gene therapy at the Great Ormond Street Hospital and University College of London Institute of Child Health in London. He trained in medicine and pediatrics before developing his T-cell gene therapy research at UCL using retroviral and lentiviral viruses. But he has led also translational application of human genome editing, including Talon's, CRISPRs, and base editors, and clinical trials that have focused on exploiting engineered T-cell against leukemia and addressing barriers to improve transplantation. I think he will discuss with us today this gene editing modification for CAR T-cell that he can use to treat patients with ALL. Professor Qasim.
Thank you, and good afternoon. I'm going to fill you in on our work with genome editing of CAR T cells to overcome barriers to allow them to be used off the shelf, in particular for pediatric leukemias. A few disclosures on that first slide, and a quick word that we are less than 1 kilometre away from here. We're part of UCL, but also part of the Children's Hospital, just a few blocks away. And our expertise over, 2 decades really, has been picking things from laboratory that can go into translational steps, in particular around engineering cells using viral vectors and editing technology, taking them through a manufacturing phase under GMP conditions, dealing with the regulators, the ethics and testing and so on, and then setting up the infrastructure to deliver advanced therapies. And our own interest is in children, but that's then been spread out to other applications as well. So for today's set of slides, I'm going to run through the feasibility and use of allogeneic CARs. I'm going to talk about trials using Talon editing, which is an earlier version of genome editing technology, in particular a product called UCAR19. I'll then talk about where we are applying newer technology using CRISPR and how that's been translated and jumped into clinical phase testing in the UK. I'll include some discussion around base editing, which is an evolved version of CRISPR editing with some advantages, and update you on where we are with translational steps and what's coming next. You've heard already the ins and outs of CAR design and TCR and so on, so I'm not going to talk about this except to say the versions we're going to talk about are second generation CARs, and what that means is all our constructs that we're describing today have got this 4-1BB and CD3 zeta domain, and the configurations are kept the same for most of our products that I'm going to show you. And again, I'm not going to spend any time going over this except to say there are a number of phase 1 trials in blood disorders for using CAR therapies. The majority have been around CAR19. There are some other indications, particularly for the B-cell malignancies. And as you've heard, half a dozen of these products are now available as licensed products. But they're all in the autologous setting at the moment. And there are some limitations around not being able to harvest enough cells or doing that in a timely manner to get your treatment back to the patient early on in their disease. There's the issue of escape and mutations arising so disease can invade the immune response that you generated. And of course, problems with cytokine release and neurotoxicity, which are generic across the applications, and a whole lot of unmet applications as well. So I'm going to talk about how we can address in particular the issue of not having enough cells or not having them quickly enough, cheaply enough to be able to deliver to your patients, and how we might be able to address that. And this brings us to discussing the difference between autologous and allogeneic approaches. And most of you will be familiar with the process around generating autologous CAR cells taken from a single patient, normally to a central manufacturing facility, engineered, released, and brought back to that patient. That process can take 3, 4, 5 weeks, depending on what the setup is, and is rather logistic heavy and quite costly. With the allogeneic approach, what we hope to do is use a single donor or a limited number of donors to generate banks of cells that can be used without HLA matching, have them pre-manufactured, frozen in aliquots and banked, ready for distribution to when and where they may be needed. Now, to achieve that, there are some important hurdles to overcome. Of course, the immune barrier is the most pressing one. There are some lessons we have from transplantation where we talk about the pyramid or the intensity of lymphodepletion that's required to overcome a HLA barrier. And in general, there's two aspects to this. One is how we prepare a patient or a host, and that generally involves chemotherapy and or radiotherapy. And at the bottom of this pyramid, there's very intensive regimens. At the top, there's some rather gentler ones or minimal intensity ones. And if we can get away with using minimal intensity for our patients, that would be advantageous. And I'll highlight this product here, Campath, which is an anti-CD52 antibody, which is very lymphodepleting, but may allow us to use reduced amounts of other agents to overcome these type of barriers. And of course, the second part of this is how we prepare the cells or we prepare the graft in the transplant setting. And in that context, there are various maneuvers that are possible to engineer grafts, a lot of them involving kit like this Miltenyi system, where we can deplete, we can select CD34 cells and leave the T cells behind, or we can deplete alpha beta T cells. And that technology is quite widely available now in European centres. So in the end, what we hope to do is end up with a product or a cell therapy that looks something like this. It's been engineered, it's been disarmed so it doesn't cause damage through allorecognition by removing the T cell receptor. It's been engineered to redirect its specificity either with a CAR or with a TCR, this example showing CAR. It may have engineering for genes like checkpoint pathways to promote persistence. And it could have had the MHC molecules removed or CD52, which allows it to evade the effects of alemtuzumab. So those are the engineering steps that have been postulated for some time to allow us to use cells off the shelf without having to match them. And the first iteration of this to reach clinic was using a reagent called Talon. We got involved in this in the around 2012 or 2013 using something called a Talon mRNA to edit two genes. One was a T cell receptor gene, one was CD52. And after some preclinical work, the first patients were treated around about 2015 using products that were generated at the Gosh site. Now, at that time, manufacturing these cells was actually quite labour-intensive and involved multiple steps of hands-on activity. It's summarised in this slide here, the state of play back in 2015, with multiple bits of equipment involved for washing cells, and a electroporator for allowing Talon mRNA to enter it, a retroviral vector transduction step, a wave bioreactor, a Clinimax depletion machine, and then at the end, a freezing step of the aliquots. That whole process was taking us between 15 and 18 days with 4 full-time people involved. Now, those products were manufactured and were used in 2015. And even at that time, we knew, although they were very effective both in our models and in the preclinical stages, we knew from our investigations there were some drawbacks to editing and multiplexing cells. And that's shown on this slide here. And if you, and as was discussed this morning actually, if you create multiplex changes in cells by breaking chromosomes, there's a likelihood of translocations. And our preclinical data was telling us that that was sitting at around about 1 in 100 to 1 in 1,000. Our GMP batches could pick up some of these changes. Here's a translocation event between 1 and 14, and we could see them on fish. And the frequency of those events was probably more than 1%. It may have been up to 5%. So we needed to contain our application, knowing that these changes were in the cells. And the first configuration of this treatment was something akin to a bridge, a journey that takes us into remission and allows us to deliver transplantation or curative transplantation by removal of the cells. So this was the proposal. We delete host cell immunity by using a combination of chemotherapy and alemtuzumab. We infuse cells for a time-limited period to allow them to remove residual leukemia. We remove the cells with additional round of chemotherapy, and then we reconstitute using an allogeneic donor. And that concept was accepted by the regulators in the UK as a safe way of applying this technology for the first time, even though the batches had some changes that we didn't really know the effects of. And the first experiences of this were published back in 2017, where the first 2 children that we treated had gone into remission and responded. That was then picked up by some commercial partners or funders to take on multi-centre studies both in the UK and internationally. So on the left, we can see this is the kind of results we would see in the successful ones, where there's disease shown by these two markers at reasonably high levels, and then they receive a round of lymphodepletion, a single round of cell infusion, enter remission, and that remission, in fact, for these two first patients is still sustained some 7 years later. The larger study in adults and in children was published in 2020, and although the efficiency rates were not as good as autologous therapies, it did offer a route to treatment for patients that lacked other therapeutic options, and those are ongoing in some forms of B-cell malignancy. The technology in the meantime was galloping ahead, and so our academic studies were starting to look at other applications even while the technology was coming through. Now, there's a number of similar trials that are up and running. The top three I've shown are the Talon versions funded by Allogene and Servier. There's a version called a meganuclease, which is being run by Precision with an inserted CAR into the TRAC locus for the same indication of B-cell leukemia. CRISPR Therapeutics have a study which is running with TRAC and beta-2M knockout, and there's other knockouts, including a PD-1 in some of those products. Our own version, which was developed on site, is something called TT52 CAR19, a mouthful, but I'll describe what we've done there and how that's faring in the first part of our clinical phase testing. So this is a universal CAR19 where the genome editing step is linked to the knockout step to allow us to produce a much more homogeneous and effective product. So let me just run through this. The technology, as you've heard this morning, is Cas-based, and we're relying on this non-homologous end joining to create a deletion of the gene. We're not doing what some of the other trials on that previous slide had shown you. We're not yet able to insert specifically into the gene, the TRAC gene. This is adding a gene using a lentiviral configuration. But that configuration has been modified somewhat to allow us to express both guides, double guides in the long terminal repeat of the vector. And the reason for designing them in this way, there were two reasons. One was to allow us to link knockout and CAR into cells. And the other was a practical logistical issue that when we set up this project a few years ago, there was no access to a clinical grade guide reagent that would have allowed us to go into clinic where we could make clinical grade lentiviral vector to do both steps. Cas is delivered transiently by mRNA through electroporation, so the effects of this enzyme are limited to a few days and then it disappears. Now on the right, you'll see some flow plots, and the key one to look at is the bottom one here. So at the end of production and processing, our CAR cells are highly expressed. They're 90% plus CAR positive, and that's consistent because of the linking of the knockout and the transduction. And there's a high percentage of double knockout for both the TCR and the CD52. Now, the other bit of technology that came along and has improved things immensely has, instead of those multiple bits of equipment and lots of hands-on time, we use this device. It's called a Prodigy. It's a German Miltenyi device, automates large percentage of the activity. And we do a stage which is off machine, off device on an electroporator to allow the entry of the Cas. And the first group of patients to receive this batch, or these batches, have been children in the UK, across the UK, who are in this situation otherwise. They'll have had a transplant or have had a previous intensive cell therapy and will be facing a prognosis where survival is actually pretty much under 20% if they in that situation after a first relapse after a first graft, both in the context of B-ALL, and I'll show you this dotted line for T-ALL because we'll come on to that as another disorder towards the end. So the phase 1 trial was approved, in fact, just before the lockdown started, and then there was some delays getting it opened, but it is open and has been for just over a year. It's treating children up to the age of 16 with either a failure or unsuitability for an autologous CAR19 product. They receive lymphodepletion with a combination of fludarabine, cyclophosphamide, and that alemtuzumab drug, anti-CD52. Single dose of cells are infused, and they're monitored in hospital for 28 days, at which point they undergo a bone marrow examination. And if remission can be achieved, they then become eligible for an allogeneic transplantation, which then removes any resisting CAR cells and allows them to proceed into a longer-term follow-up. Now, it's a phase 1 study, so the key outcome measures include tracking of toxicity and safety in the context of children receiving the treatment. And in particular, we point out CRS occurred in all the patients, but was manageable at less than grade 2. There was one significant neurotoxicity which resolved spontaneously. Viral infections and fungal infections were monitored carefully because we do worry about the effects of lymphodepletion, and cytopenias because of the consequences of the augmented lymphodepletion that we use, all documented. So that study is open and recruiting, and will continue to recruit for the remainder of the cohort. In the meantime, as I mentioned, the technology hasn't stood still. So we have an additional piece of technology that has evolved from CRISPR, but allows us to edit cells without causing double-stranded breaks by using an enzymatic method to convert residues, base pairs, to allow us to introduce, in particular, stop codons. So this has come out of MIT from David Liu's group in end of 2016, early 2017. And there's 2 iterations of this. It's the top one I'm going to talk about. It's a cytosine deaminase enzyme fused to a deactivated Cas that drags the enzyme to a very specific point in the genome and allows us to carefully deaminate cytosine residues. You have to go back and look up A-level biochemistry, but cytosine, if you remove the amine, ends up as a uridine, and that, as cells are replicating and repairing, is recognized as a thymidine. So it's a C to T conversion, and if we use that in this first instance, we can create stop codons within almost any gene we like, and the ones we've picked are the same ones that we're using in the other trials. So this is a way of creating a stop without a break, or disrupting a splice site so we don't get a proper gene expressed. And that now allows us to edit genes without the translocations. Now, there's all sorts of other applications in mind for this technology in terms of repairing inherited gene disorders, but this is a nice place to start because it's about breaking things rather than repairing them and making sure everything else is okay. This is a good place to start. Now, the technology itself is involving these conversions, and there may be other stuff going on that we have to think about. These are guide-dependent effects, meaning there's a guide that's taking the enzyme to a specific locus. There may be guide-dependent off-target effects if the guide is recognizing other sites. There may be other effects within the window of activity that we need to be concerned about. There may be guide-independent effects or promiscuity that we need to be concerned about, both at the DNA and at the RNA level. Nonetheless, the technology has turned out to be incredibly efficient, and these are some examples of our testing of editing of the TCR beta locus this time, where we get 100% conversion at the site of editing, and all the activity is in a 5 base pair window as advertised by the original version of the technology. And we can do that across multiple target loci in the same cell without causing translocation issues. And that translocation issue is shown on the next slide, where we compare the activity of a Cas enzyme which causes cuts and an editor which doesn't cause cuts. Again, 2 loci that have been edited in this example, and then compared side by side using digital droplet PCR. And we see, as previously we mentioned, there's up to 1% of translocations being detected, predicted translocations being detected with Cas9, and those are eliminated with the introduction of the base editor. So where to apply this technology for a first-in-human application? Well, I mentioned on an earlier slide that as well as B-cell leukemias, we see T-cell leukemias, but there's never really been a good CAR T-cell therapy against T-cells because of the issue of fratricide and cells killing each other during the production phase. And there are a number of targets for T-cell leukemia which are being investigated either with antibodies or by CAR therapies. And out of these, CD7 is the one that I'm highlighting on this chart here as the one that's being pushed forward for phase 1 testing. And there's some interesting studies coming out of the Far East at the moment where that very same approach using another technology to remove CD7 called restriction technology, protein restriction technology, has treated a number of patients with some rather nice results published last year of remissions of T-cell leukemias both in children and in adults. So encouraged by that, we proposed a phase 1 trial in children of an allogeneic off-the-shelf product, which would have 3 edits. One edit would remove the T-cell receptor. The second would remove CD7, and that would stop the fratricide. The third would remove CD52, and that would allow them to operate in the presence of alemtuzumab, much as we did for the first study. So that has entered manufacturing phase during the last 2 years, and using the same types of equipment and technology we'd already set up for the CRISPR trials. So there's a prodigy involved, there's an electroporator. This time there are guides, RNA guides that have been manufactured at high quality, and there's an optimized editor mRNA that's supplied by BioNTech, the same company that makes the vaccine. And those are used to engineer healthy donor T cells collected from volunteers arranged by the Nolan registry. They're edited, they're transduced, remaining TCR alpha beta cells are depleted using the Clinimax system, and then they're frozen in dose aliquots. And then these cells go through the same type of testing that I showed you for the B-cell leukemias. I'm not going to run through all of that. I'm just going to add a couple of extra points that we screen for on and off target activity at the 3 sites that we've edited using at sites of predicted activity using next generation sequencing. And we undertake a crude form of promiscuity screening by seeing if there's been any modification either in RNA or DNA in the sequences encoded by the binder regions of our CAR, because we figured if there was promiscuous activity and it occurred here, it might redirect the CAR to another site. But in fact, we don't see any. So the sea of blue means there's nothing much going on, and we're reassured by that as far as it can. So again, a phase 1 study is now open and recruiting at GOSH. This is the first application of a base-edited CAR T-cell product. It's recruiting from across the UK for children up to the age of 16 who have relapsed or refractory TLL and have exhausted other therapeutic options. And it's going to follow a very similar structure to the previous studies, where there's some disease reduction using standard of care therapies, lymphodepletion using that same combination of fludarabine, cyclophosphamide, and alemtuzumab, a single dose of CARs with a dose extrapolated from other people's published studies. It's going to be just under the 1 million per kilogram dose range. 28 days of hospitalization is expected. And then if there's remission, there's an additional round of reduced intensity conditioning and then transplantation to re-establish the T-cell compartment, which of course, unlike B-cell aplasia, is not compatible with survival. And so that study is open and recruiting and is underway at GOSH. And that brings us to where we're heading for the next couple of bits of this. I'll summarize them on this slide here, that the phase 1 CRISPR-Cas9 is open and running, the TLL is running, and in fact, this study is linked quite closely to a second one funded by Wellcome, which is called Deep Conditioning for AML, where there will be a combination of CAR T cells off the shelf infused against multiple targets to try and secure remission in those patients ahead of an allogeneic procedure. So that we expect, perhaps, if not the end of this year, sometime next year should open for recruitment. Okay, so I've got a summary slide. So in summary, genome editing, I think it does answer some of the hurdles and issues we've uncovered in the autologous setting, but there's quite a lot of work to do to unravel where the applications will be most effective. The platforms are evolving faster than we can keep up. So whatever we're using today may not be what we're going to be using in 5 or 10 years' time. But that's okay. For a phase 1 setting, it's ideal and it allows us to compare different platforms for these applications. And of course, there will be this important issue of how we monitor and keep track in the longer term to assemble safety data that can be spread across for other applications. That's me, and a summary slide with a large number of people involved, not just within the group. I'll highlight the GMP team, Annie Yotek, Hong Zhang, and the preclinical people who've done a lot of the work, Christos and Roland and others included at our site, but also at King's and wider at UCL and at GOSH. Thank you.